Everyone, and good morning, guest speakers. On behalf of organizer, I'd like to welcome all guest speakers to our session, as well as everyone who are attending our session this morning. This morning, we have 6 guest speakers in total and 1 professor from UBC who will give us a session, a closing remark. 6 guest speakers will present their views on bamboo forest biodiversity conservation, ecosystem, adaptive management and sustainable development, under the vision of carbon neutrality. I will host the first half of the session, and which includes 3 guest speakers. Dr. Luan Junwei will host the second half and the closing remark. Each guest speaker will have about 12 minutes to present, 12 minutes. Please remind you, kindly remember that, stay on track. Q&A session will be held after the closing ceremony if we do have some time left. We truly appreciate if you are not presenting, please kindly mute your microphone during the whole session. Thank you so much. Now, let's begin our session. Next, let's welcome our first guest speaker, Dr. Hu Yonghong, who is President and CEO from Shanghai Chenshan Botanical Garden. The topic is the important role of plant carbon sink capacity on sustainable development. Please welcome Dr. Hu Yonghong. Welcome.
Dear colleagues, good morning. Many thanks for Chairman and the invitation. Congratulations for the INBAR's 25th anniversary. My name's Hu Yonghong from Shanghai Chenshan Botanical Garden. And my topic is to calculate the carbon sink accurately in the city, so we can understand it's very important on the background, like how to make the carbon reduced and increase the better life. So we start from the plants. The plants, the most important and fundamental function is to balance the carbon and oxygen, especially by the photosynthesis. So the carbon was fixed in the plants and the organic matter was produced. So it does the same from, in the urban areas, the plants do the same function.
Dr. Hu, sorry, your PPT is not showing on the screen.
Sorry.
I'm so sorry.
Sorry, I just...
Okay, great. Now we can see.
You can see that? Okay.
Yes, we can see, but you have to enlarge it.
Okay. It's good. Okay now?
No, it's gone. So maybe you just keep as it was. Yeah, but only half of it. We only see half of it now.
I think some problem for the, for the system, the.
Okay.
Something wrong with my system.
Okay. If you cannot enlarge it, it's just keep it as it's okay.
Okay.
Yeah, it's okay. We can see it now. What you can do is you have to enlarge it. Can you do? Yes, great. Yes, wonderful, wonderful. It's on. Yes.
Because not I cannot operate. Maybe someone can help me for the operation.
Oh, you cannot operate. Can you?
It's by the So we can next, and so I can follow. Continue.
Okay, great.
My talk. Okay.
Yes,
Please,
great.
Next.
Okay, next.
Sorry for that.
That's okay. Yeah. Okay.
No, it's okay. You know, the plants in the is very important. And next, please.
Next. Yes.
So next.
Okay, next. Okay.
But you know, the performance plants in the urban habitat is quite different from nature because of the, you know, the limited growing space and the very harsh soils, and so it's caused the very poor growth. So next, please. And so we cannot find the way to calculate the carbon fixation. So because, you know, it's quite different from nature, and also because the, you know, the artificial disturbance. So next, please. So for the existing modeling for the, you know, for the calculation is not suitable for the urban plants, and we don't have the database. We have that in nature, but we don't have it in the urban areas. So, and many ornamental plants in the cities, their biomass model are missing. We don't have that. So next, please. So the objective is to aim to calculate accurately for the carbon sink of urban plants by the new way, or maybe we say that's by accurate way. So next, please. So we did the field investigation and analyze in 9 cities, and with 1,500 plots, and to test the 353 species in these areas. Next, please. And we do the biomass measurement, and with regression, we cut all the plants and calculate the biomass. Please, next. Next, please. So, and also we do the photosynthesis, and we compare the two methods. Next, please. Next, yeah. And we assess the typical communities, the structure. Next. And also we did the design for the high carbon fixation communities. Next, please. So we can find that the plant used in the city is very little. You know, the only 16 plants are very commonly used. So first is Cinnamon camphora is 44%, and the second is 22%. So this you can see that the plants is not very diverse. Next, please. And we do the biomass regression by cutting the plants and calculate. So we adjust the coefficient. Please, next. And also you can see that the net photosynthetic rate for 300 plants that are very concentrated from 3 to 3.8 to 8.8 altogether. And the net NPPs, net photosis rate, is from 3 to 8. So next, please. And we can also understand that the deciduous shrubs have the very high NPP, net photosis rate, and the second is for the evergreen trees. Next, please. And also we found some plants with very high carbon fixation per areas, per square centimeters. So you can see it some figures, and Celtis and Nelumbo, and also Sapium. These plants with very high, the per square centimeters, very high carbon fixation. So next, please.
Oh, you only have 2 more minutes to go,
Okay.
Dr. Hu. Sorry about that.
Okay, no problem. So also you can see that the individuals are quite very high, with a very high too. Next, please. So that we can find the result for each individuals. Next, please. And we compare and find the the fitting degrees reached 87%. It's more than the other did. Okay, next, please. Also, we transfer the regression from LAI. It's like the total leaf areas to plant traits like the height, current diameter, and the DBH. Next, please. And also we do the automatic calculation by remote sensing images based on the current diameter and the heights, the crown heights. Next, please. And also we do the living vegetation volume with the biomass, the whole plants with the biomass by the remote sensing. Next, please. Also, we do the classification and recognition of tree species, and also automatic measure it by orange crown diameters. Yes, the same. And next, please. So also we do some the communities. Next, please. So we recommend the 15 community configuration models for so they can fix 10 for each hectare each year. So it's very high number. So next, please. In the end, I think that the conclusion is we can find the 10 native biomass regression of the common woody species. So it's to build the new models. And the second is we found the non-destructive models for the single plants. So it's very accurate with the accuracy rate is reached 87%. And also we use the remote sensing to recognize plants and to calculate the carbon fixation by in the heterogeneous urban green species. So it proposed 15 community configuration was used, which can the capacity fixation more than 10 each hectare each year. Next, please. So some suggestions we can suggest more plants was used in the future with a very high carbon fixation. Next, please. And so enrich the plant configuration arrangement, maybe more professional arrangement. Next, please. And in the end, we try to suggest to improve the urban soil quality to increase the growth of the biomass plants. In the end, we can fix more carbon dioxide. So it's my talk. Thanks for your listening, and your question be appreciated. Thank you very much. Sorry for the system problem.
Thank you, Dr. Hu. That was a fascinating presentation. We all learned about the plant carbon sink capacity plays a very crucial role in sustainable development. Thank you again. So thank you, Dr. Hu. And I think, thank you for your presentation. Next, let's welcome our second guest speaker, Dr. Denson Warreru. Welcome, please, who is from Foster Department of Water, Irrigation, Environment, and Climate Change, Nairi County Government of Nairi. The topic is a role played by bamboo roots in substrate growth and the life enhancement of aquatic life. Welcome, Denson. The floor is yours. Mr. Danson, please share your screen. Mr. Danson, can you hear us?
Yes, I can.
Great. The floor is yours. Please start your presentation. Thank you. Can you hear us?
Yes, I can.
Oh, you can. Okay, great. Okay. Can you please share your presentation on screen?
Yes.
Great.
One.
It's coming. It's coming. Yeah. Wonderful.
Yes.
Yes. Ms. Danson, you only have 12 minutes to go. Thank you.
Good morning, everybody.
Good morning. Good morning.
This morning it's at 3:30, 3:35. I am done somewhere. It's my pleasure to participate in the second global Babu and Rattan Congress with a very nice presentation on low paid by Babu foods in substrate growth and life enhancement for aquatic life.
Mr. Danson, I'm sorry. Can you please speak a little bit louder because we cannot hear you?
Oh, sorry.
We cannot hear you properly. I'm so sorry. Can you please speak louder?
Hello, can you hear me out?
Yeah, it's better. It's better. Thank you.
Oh, okay.
Great,
Sorry.
great.
I think
It's much better now. Yes.
okay.
Please go on.
Yes. And now I proceed on a brief introduction on Babu as being a purifier for the runoff that might be harmful to the any harmful material in the runoff, mostly in the water that comes from towns, that comes from areas that we farm. By this, it is able to remove the impurities and pollutants that by use of its network of rhizomes that is very effective. That's a process known as phytoremediation, the process of using the plant matter to filter water and remove organic and inorganic contaminants. The plant tissue of Babu absorbs contaminants, water, and removes, stabilizes, or destroys the harmful substance. Can we proceed, please, to the next slide? Can we proceed? Sorry. Yes. In the role played by Babu's roots in food growth for aquatic life, we start by the commensalistic relationship between Babu and phytoplanktons and phytons. In this, Babu is grown on wetlands, is known to have a commensalistic relationship between the phytoplanktons and periphytons. The growing of Babu in the environment has beneficial aspects together. The growing of Babu together with the periphytons and phytoplanktons has known to have the beneficial aspects. Babu has recently been shown to be the growth substrate. Sorry, exit this. Babu have recently been shown to provide suitable substrate in periphyton-based aquaculture system, which enhance fish production in aquatic ecosystem through periphyton and associated microfauna. the stats of babu can positively affect the aquatic life for periphyton community because they provide a cooling effect to the environment. The reefs are known to insidiate the water reservoirs and experience no or little allelopathic conditions. In aquatic life, there exists cyanobacteria that produce toxic compounds that have harmful effects to aquatic life and terrestrial animals. The blooms of the cyanobacteria in aquatic system are as human consequence, that is, as a result of eutrophication caused by human activities, all, in other words, the anthropogenic activities that are a major environmental issue. The bloom can eliminate plants and aerobic animals in the ecosystem by covering the water surface and depletion of oxygen. Babu enhance the growth of competitors that inhibit cyanobacterial colonies. Notably, babu contains high concentration of silica, which is an essential nutrient for algae, especially diatoms and babu starts that accumulate and exports downstream a high quantity of silica generated by logs weathering. The stimulating of these will now act as a competitor that will outcompete the cyanobacteria, being a very effective biological control of cyanobacterial blooms. Besides, the babu roots are known to have a bioagent that protects the roots of the babu from destructive fungus. And in this now, it's shown to protect itself from now any destructive microbes or fungus. And a case study in Nyeri County, we did on Nairobi River in Nyeri County. It has been observed that there is high quantity of life in areas with babu. The growth of buffer flora and fauna along river has exponentially increased due to increased babu growing, a clear indication that babu has played a role in life enhancement. Another topic on subtopic on growing of babu, we have babu can be grown without chemicals and pesticide. This shows it can be grown in 100% organic, can be grown 100% organically. This is why babu is recommended for riparian conservation of, or in our case, the riverine planting. By virtue, babu is grown organically, it maintains the pH of the environment at alkaline. This again is an eco-friendly property of the babu plant. The aquatic life is not exposed to chemicals and fertilizers. That's when aquatic life, especially fish, eat phytoplanktons and periphytons. They take in form of alkaline nature, contrary to the absence of the babu plant. We have a figure, a pictorial representation of babu glowing along the river. We have diagrammatic representation, of how the babu is able to support the growth of the competitors of cyanobacteria in port, river, or lake ecosystem. You can see now the stats of babu along the river. We are able to see that it is able to exudate the dissolved organic matter, silica, that will stimulate the growth of diatoms, green algae, and now these will act or invade or outcompete or compete the cyanobacteria. And now we go to the second topic on phytoremediation, babu as a purifier. And babu is known to act as a buffer for purifying the runoffs that will be harmful, that are mostly harmful to organisms in water. It has very high dense root system, resistant to very many environmental stresses. This is able to remove impurities, pollutants from the runoff, cushioning the life in the water. The dense...
Mr. Dance, I'm sorry. Kind
Yes.
reminder, you only have 2 more minutes to go. Thank you. Sorry
Okay,
about
okay,
that.
okay.
Sorry about that.
Thank you. By this, it's able to remove the impurity, then we are able to see now the root rhizomes, and we have been able to plant to rehabilitate rivers, that is Nairobi River and Chania, and by the in both rivers we are able to see that we have informal settlements along the river. We have towns that are known to drain water that is harmful to the ecosystem. And in one of the cases in Nairobi River, we have one of the environmentalists who has been able to plant bamboo along the river, that is Nairobi River, and it's able to, we have been able to clean and to actually protect the river. We proceed. Now you allow me now to proceed in a hurry. You can see through. It was observed that fish exponentially increases. That is Nairobi. We have these rivers. We have figured out Nairobi River. We have this subcatchment that we observed that fish increases exponentially as bamboo increases. With and now we have this one here. This one of the videos in that you can be able to see that water is very clean by use of bamboo. And thank you guys for your kind attention that we have been able to meet and work on these issues. In nearly we have been able to work on these issues. I have been able to proceed with these issues of looking through and doing my research on these areas that are very noble. I hope one of the guys comes across and we will work this out together, because we have some shortcomings in some areas, and we hope this will be a success in future. Thank you.
Thank you, Dr. Danson Variro, and thank you for your excellent presentation. We all learned the importance of bamboo roots in the substrate growth, and we also know the bamboo is a great purifier for our environment. I think your research is fascinating. Yeah, thank you. Thank you.
Yes. Welcome.
Yeah.
Yes, welcome.
Okay. Thank you. Thank you so much. Now let's welcome our third guest speaker, Mr. Huo Xiaowei, who is a Vice President of Beijing Tsinghua Tongren Urban Planning and Design Institute. His topic is a coupling of human settlement and biological diversity, and also practice and thinking of culture, landscape conservation. Welcome, Mr. Huo. The floor is yours, please. Thank you.
能看到我的屏幕吗？
能
Can
看见。
everyone see my screen? Yes.
谢谢主席，大家好。
Thank you, Chairman. I'm very glad to attend the second Global Bamboo and Rattan Congress to introduce the cultural landscape consideration. The Institute INBAR have cared about the human settlement. Human settlement actually is a very extensive topic. It has a connection with many other disciplines, including the natural environment. Our institution actually have cared about life and diversity, as well as a relation between the ecosystem in the urban cities. That is the report we have. Action plan we have released in 2012. In our research, we understand that the diversity research actually is not only for environmental protection, but it's also for us to live a better life. So the diversity protection in human settlement need to start from the relationship between human and nature. And many good cases actually the interaction is a result of interaction between human and nature. So we call it cultural landscape. The cultural landscape by UNESCO, one of the definition from UNESCO actually is cultural landscape. And also it's further divided into 3 types. One is the clearly defined landscape. Second is gradually evolved and associated cultural landscape.
强调人类与环境之间的相互影响以及整体保护的理念，突出了自然遗产与文化遗产从2元走向统一。我们今天说的很多文化景观遗产，不仅属于过去，其实它是人类与自然共同创造的人居环境的优秀代表。在文化景观遗产所在的地区呢，呈现出非常美好的人居环境，人工的建筑与自然相互映衬，成为一个整体。在这样的环境中，生物多样性也是非常突出的。比如杭州西湖区域内的各种物种达到1200多种，尤其是各种鸟。西湖的10个景观中有非常著名的一个叫做柳浪闻莺。五台山也是世界文化遗产，是中国的4大佛教名山之首，保存着世界最庞大的佛教古建筑群。它将佛教的崇拜凝结在对自然山体的崇拜中。独特的地理环境也形成了五台山同样非常突出的生物多样性。比如这里狐狸很多，遇到狐狸会被认为是吉祥的象征。IUCN 1991年提出，地球上每一个物种都是所有生物体构成的生命群落的一部分。这些群落将所有的人类社会，包括现代和将来的人类连接在一起，将人类与自然界的其他部分连接在一起，既包含了生物多样性，也包含了文化多样性。文化与自然融合的观念正在越来越成为国际的共识。有很多中国的学者很早就提出对于自然与文化融合的观念，比如历史学家钱穆就提出天人合一这个中国的价值观一定会对于世界做出贡献。物理学家钱学森也主张要将城市建设与自然环境相融合。下面呢，我介绍2个我们实际参加的案例，一个是云南的景迈山古茶林文化景观申报世界文化遗产。景迈山古茶林是中国茶文化景观中的一种，是目前世界上保存最完好、年代最久远、茶景观最典型，也是人地关系最和谐的古茶林，被称为人类茶文化史上的奇迹。左边的这个就是古茶林。古茶林呢，跟一般的台地茶林具有非常不同的景观，它远看是森林，近看是茶林，这取决于当地独特的种植技术与传统。景迈山古茶林文化景观位于云南省的普洱市的景迈山上。大概公元10世纪，布朗族的先民在迁徙途中发现了野生茶树，他们在景迈山的森林中建寨子，在寨子的周围栽培茶树，并探索出利用森林的生态系统，在高大的乔木下种植茶树的技术。然后呢，与其他民族一起，历经千年，形成了林和茶互生、人地共融的古茶林文化景观。从图中呢，大家可以看到土地的塑像例证来看，景迈山呢以古茶林为核心，生产、生活和生态用地空间合理分布。山顶呢是神山所在，也是各村寨的水源保护。那么古茶林和村落呢，在山地的古茶林的外部还有一些防护。生产粮食和蔬菜的耕地呢，是在海拔相对较低、水源也充足的地区，避免种植的过程中对古茶林的干扰。景迈山的先民呢，在天然林中砍除了部分的乔灌木，留有一定的乔木来遮阴，为茶树创造了良好的生长条件，并且呢，有选择性的保留，比如说香樟树，也有选择性的清除一些会与茶树抢夺水分养料的芭蕉，从而可持续的生产出高质量的有机茶叶。在古茶树上还有非常独特的寄生植物与昆虫。古茶林呢，由布朗族和傣族等村庄居民长期种植和管理形成。这些传统文化体现在生活的很多方面。几百年前的古塔的雕塑上面呢，就有茶树的形象，这在塔的雕塑中是不多见的。
trees
庙宇
in
的木
the
雕中呢，
sculpture,
也有
which is
非
kind
常
of
丰富的
rare.
生物形象，花、
And
鸟、
here
虫
you
兽，
can see in
它
the
们与人
sculpture
的
there are
形象
many
是平等
birds,
的。这表明了布朗
grass,
族居民
and which are
万
the
物有
same
灵
or
的观
equally
念，
important as
人
the
对自然
people.
环
We
境
can
充满
see
了敬畏
every
与喜爱。
living being in the world
在
is
村落
equal,
的
and
佛寺
people
中
really
种
admire
植
this
着南传
nature.
佛教中具有神圣意义
Birds
的
and
5种树和
animals.
花，此
There
外
are
还
some
种植
very
着一棵
important
象征着茶的
trees
纯
and
纯的
flowers
茶神
in
树。
related to
从
Buddhism
遗产的
and
价值阐
tea
述中可以看
trees.
到，当地人民利用
You
地
can
方
see
的
the
森林
cultural
生态
landscape
系统，
value of
创造性
Jinkai
的
tea
发展出
forest.
茶种植的
The
文化
local
系统，
people, they
并形成
develop
了以
the
茶叶为
culture
主的生产
the
生活
tea
方式，
plantation
实现了
and
人
also
与自然的
incorporate their
长期
lifestyle.
和谐的互动。古茶林的
Create a
遗产
very
要素呢是
benign
非常复
interaction
合的，
between man
包
and
括5
nature.
片古茶林、
The heritage
3片防护林，
culture,
以及9个传统村落，以及
and
村
there
民
is
们的
some
茶文
the
化、
ancient tea
茶种植的技
forest,
术、种茶、
traditional
制茶的技术、
villages,
民
the
居
tea
营造的技
making
术等等传统民俗。
techniques, and also living some
对
folk
景迈山古茶林的保护理念
customers.
中呢，
The heritage
最核心的
conservation
就是对于这
concept
些当
of
地居民
this
生产生
is
活传统的保护，让
the
他们能够
protection
继
of the
续
people's
以
way
茶
of
为
life.
生，能够拥有美好的家
They
园。
can enjoy
所以，
their
首先
life,
是建立起遗产
and
的保护条例，还有
the first set
村寨
some
的一些
heritage
规
conservation
定，
management
加
plan
强对于
and
传统
rules
文化的保
and
护。
regulation for villages and
那么
villagers.
传统民居中呢，大家看
The
到呢，从历史上
traditional
看也不是一成不
dwellings,
变的，
they
但
are
是呢，
also
需要
evolving,
引导民居的新建行为，避免传统民
and
居的
we
疏
need
裂。
to protect
所以呢，也实施
these
了对
Asian
传统村落中
architectures,
民居
and
建筑以及建筑的
here
环
we
境的
have the
改善，对基
implication
础设施的提
implementation of
升。
the traditional village
传统
conservation
民居修
projects
缮之后呢，
improve
就更加的
their
安全舒适，
the
人
living
更愿意居
conditions
住。
for the local
村落
people,
中的环
and
境
they
呢也
are
很
very
整洁，
popular
生态。
after restoration. It is very
民居
clean.
建筑与环境很好的结合在一起，
That is the integration
室
of
内也
the
更加的舒适。
architecture with the nature. I can see the
在村
inner
庄内呢，
side
还
of
开设
the
了一处介绍
buildings.
茶文化传统的
This
展览馆，
is a
讲述
museum
地
of
方的
tea
生物
culture,
多样性，
which
吸引不同
is
背景
about
的
the
人来这里，
biodiversity of the
来这
village.
里的
This
参观学
museum
习，
can
更深入
attract
的了解
many
古茶林的
visitors
知识与
to
文化，
know more about the
形
tea
成更为美
culture
好的人居景观。
and to create
那
some
么让当地
very
村民更
beautiful
加稳定
cultural
的生活在这
landscapes.
里。那么
Villages,
形成一个持续的
people
一
in the
个茶为
village
生
are
的一个社
enjoying a
区。
very
那么还有一
happy
个例子呢，
life.
是关于
Another
在
example
江西鄱
is
阳湖
the
畔的鲤鱼洲
Li
的农场，
Youzhou
通过
of
生态环境的改
Nanchang
善，
City in
创
Jiangxi
造人鸟共
province.
生的白鹤天堂。
The White
鄱阳湖农
Crane
场呢，位于南昌
Paradise.
市的东北，
This fish
东临
farm
鄱
is
阳湖，
located in
这
the
里有非常丰富的
northeast of
人文资源。
Jiangxi
清
province.
华大
Here
学
you can
和北
see
京大
many
学在这里开设过农
resources.
场，
Tsinghua
自然
University
环境
and
资源呢
Beijing
也非常的丰
University
富。
had
因为
a
鄱阳湖呢是东亚澳大利亚水鸟迁飞路线上
farm
不可
here,
缺少的越冬
and
地的
Poyang
中转站，
Lake is
所以每年
a stop
有
for the
60万的候
migrant
鸟在这里
birds
越冬，其中
between
白鹤数量
East
占世
Asia
界
and
总数的
Australia.
98%，
White
而鲤鱼洲的
crane is
白鹤呢又
dominant
占到
migrant
30%。
bird.
但是在鲤鱼洲，人与自然的
In
关
this
系并非
place,
一
you
成
can
不变的，
see
而是
the
经历了
relationship
阶
between
段。
people and
从
nature,
当地
which
古代的
is
庙宇
evolving.
讲述人要
Since
顺从
ancient
自然，
time,
否则
ancient
就
the
会有灾
types
祸的故事。
temple
到
which
1960年代，
advocates
人们维护造成
the
的破坏
harmony
自然。
between
再到今天呢，
man
人
and
们尊
nature,
重和
and
保
the
护自然，
people
喜
should
欢白
be
鹤，希望
obedient
能够留
to
住白
the
鹤。
nature.
经过维
And
护稻
now
田的景观，已经
more
不是
efforts
当
are
年
put
在
to
古文《滕王
protect the
阁序》里面
environment,
描述的
to
客
protect
厅府
the
主
white
从岛屿
cranes.
迎回的景观了。右下
Here
角是
also there's
50年
some
代的航拍图
poems，
大概
about
是那个时候的样子。
the white
因
crane.
此，首
That
先
is
要
a
做的
picture
是对水环境的改
before.
善，
And
恢
also
复
we
部分
need
的河
to
道，拓宽部分的
restore
水渠，
the
营造适合
ecological
生物生活的水
environment
体。
to
研究
create
白鹤的生
good
物
water
习性，
quality
比如
for the
水
white
深小
cranes.
于30
We just
厘米的水
study
体
the
最适
habits
合
of
白鹤
white
觅食。
crane. We know
根据
that
生态多样性的敏感性的分析呢，
some according to the assessment for
对
the
这一区域进行地
habitat
形和水体的改良，
of the
控
wine
制
cranes
水
and
位与
some
坡度，
efforts to
包
be
括白
made
鹤在内的
to
不同鸟
improve the
类提
quality,
供
to
适宜
improve
的生
the
计。
environment, and
人的
to
耕作行为与鸟
provide a
又形
better
成了新的
habitat
文
for
化
the
景
wild
观。
cranes.
生
This
态
is a
化低
new
干扰的观鸟
landscape,
设施是
like
人
the
与鸟和谐
coexistence
共存的
between
表现。
man
周围
and
的环境也
wild
得到很好
cranes.
的提升，
The
会为
environment
当地
has
居
been
民提供很
improved,
好的与自然
and
接
the
近的机
local
会。
people have the
我们改造了
opportunity
一
to
座
get
建
close
筑
to nature.
为白鹤共生馆，我很快
Now,
讲
this
述
is
这个与
a
白鹤
wild
相关的知
crane
识，传播人与
museum.
自然和
This
谐
is
共生的理
about
念。
the
那么
coexistence
最后呢
between
是
man
几点
and
体会。
nature.
第一
Finally,
就是
some
围绕生物
thoughts.
多样性的愿景，
Firstly, we need
人
to
类与自
focus
然
on
和
the
谐共生，以
biodiversity
人为核心，以保
vision
护
of
自然为基础，
harmonious
积极
existence
思考
between man
生
and
境修
nature,
复对
and
于
we
人
need
居环境
to
的意义，促进二者的结合。
consider
那么
the
第二呢，以文化景观的视角来认识
natural
人类的行
protection
为与
and
自然变
human
迁之间的
existence.
互动，
Secondly, we
它是一
understand
个有机
interaction
演进的
between
过程。
human
第
behavior
三个
and
是
nature
积极探
changes.
索人居
Thirdly,
环
we
境提
need
升
to
中生物多
explore
样性的策略与实
biodiversity
现。
strategies
第
and
四个呢，
practices for
就是
improving
地
the
方社
human
区的
settlements.
生活方式
Fourthly,
同生物
the
资源
traditional
有着
lifestyle
密
of
切的依
the
存关
indigenous
系。
and
这
local
样的人居
communities
环
has a
境呢，不
traditional
仅
and
仅属于历
closely
史，也应该
tended
属于未来。
relationship with
那么这
the
就是我今天
source.
想跟
It's
大家
not
分享的，
just
谢谢大家。
for today, but also for the future. Thank you.
Mr. Hall. That
非
was
常感谢
fascinating presentation.
非常精彩的一个演讲。我们
The balance between man and nature. I think your presentation and made us to think and also learned the importance of biodiversity on human beings settlement. Thank you so much for your presentation. Thank you, Mr. Hall.
谢谢主席。
谢谢. Thank you. I think the first half of the session just become the end now. So if we do have time, and maybe we'll have a 2 to 5 minutes coffee break, then I'm going to hand over to next host, Mr. Ranjun Wei. He will host the second half of our session. Thank you so much for 3 guest speakers. You had a wonderful presentation today. Thank you.
Okay. Thanks to Dr. Peng for her moderating on the first section of our session. I'm Junwei Luan from International Center for Bamboo and Rattan. I'm going to moderate the next section of our session. Okay. The first speaker of our next half session would be Professor Akifumi Makita from Akita Prefecture University of Japan. And Professor Makita made a lot of research and interesting research on bamboo flowering. So he will share his research with us about the implications of rhizome systems of bamboos for growth mode, clonal structure, and evolution of flowering behavior. Okay, Professor Makita, go ahead. Yeah.
Yes, thank you very much for introduction. Just a moment. Okay, can you hear me?
Yes. Very clear.
Okay, thank you. I'm Akifumi Makita from Japan.
Hold on, please. Hold on, please. Oh, yeah.
You present your presentation in another way round. You need to put the presenter mode.
Oh yeah, this is the presenter mode. Yeah, you can you change into the whole.
Is this some trouble?
Yes, it's in
your
the presenter mode. It's presenter mode. You need to change to the presentation mode, not presenter mode.
Sorry, I can't understand.
I mean, your presentation right now is the presenter mode. So the listeners will see the first slide and the next slide. So maybe you can, if you know how to, It's Japanese. Okay.
It's okay?
Yeah, great.
Okay.
Yeah, perfect.
Thank you. I'm Akifumi Makita from Japan. It's my great pleasure to present here, and I'd like to express many thanks to the organizers for inviting me to this session. Today, my talk is on the basic ecology of bamboos. As you know, bamboo have a wide variety in various aspects and size, shape, growth form, distribution, longevity, flowering behavior, and so on. Among such wide variety, rhizome systems make up the fundamental structure of bamboos. McClure proposed basic rhizome system, Pachymoor and Lepmoor rhizome systems. Pachymoor is a clump-forming type distributed in tropical region, and the Lepmoor type is running horizontally in mainly temperate region of East Asia. Of course, there are many intermediate types. For example, a long neck Pachymoor of Americana, Baxsphera, like this. And another is a mixed type of Pseudostachys This type have both clump-forming type and running type rhizomes in neck plants. Today, I present 3 topics to explain the importance of the rhizome system for bamboos. First one is the difference in the growth rate and the mechanism of the clump density regulation in seedling populations after mass flowering between the species with different rhizome system. And next, I point out clonal structure determining the genetic interminglement, and at last, I propose joint evolutionary hypothesis between rhizome system and the flowering behavior. Okay, go on the first topic. At first, I introduce my earliest studies that led me to focus on the rhizome systems. I made the long-term research on the population dynamics of seedling after mass flowering of the 2 species with different rhizome systems. First one is Succulensis with mixed type rhizome systems flowered in 1979 in the northern part of Japan. And the next is Sasatsuboyana with Letmo rhizome system flowered in 1977. And the brown part of the mountain flower indicates the flowered area. These 2 species show the quite different clonal growth in the initial growth stage. For Sasatsuboyana, rhizomes were developed from early stage of seedling growth, and they have many cones from rhizomes. On the other hand, Succulensis grow higher and higher in the first stage of seedling growth, and after about 10 years, they began to horizontal rhizomes like this. Growth pattern was quite different, but the change in culm density is very similar. In both species, at first, culm density increased in a few years and then decreased gradually in both species. And these figures indicate the time trajectory between culm density and weight per culm. Culm density increased in the first stage, and they became the full density state. The culm density decreased. So the overall change in the change of the population is very similar, but the mechanism for regulating culm density was different. For example, Succulensis. For Succulensis, genetic density decreased after the peak of the culm density. Seedling density decreased, which indicates the decrease in culm density was caused by self-thinning or the death of genets. On the other hand, in the case of Sasatsuboyana was quite different. This figure shows the change in bud utility ratio. Bud utility ratio is calculated the number of nodes with culm divided by the total number of nodes of rhizomes. Many culm was produced from rhizomes in the first stage after they developed horizontal rhizomes, but after that, culm production ratio decreased drastically, which means the culm density was regulated intraclonally. This result indicates the mode of interclonal competition was quite different in these 2 species. For succulents, shorter genet were shaded by adjacent higher genet, taller genet, which induced the mortality of genet. On the other hand, horizontal developed Sassatsuboyana, genet mortality may be rare and calm density was regulated intracoronary. And such results suggest the population dynamics of bamboos are greatly affected by coronal growth. And next, I'd like to briefly point out the coronal structure determines the genet interminglement. For Pacmorph tropical bamboos, genets are isolated each other. On the other hand, rhizomes of rhizomes bamboos in temperate region, genets are intermingled each other. So complicated structure. For example, most of the clump of the tropical bamboos were composed of only one clone, only one genet. On the other hand, temperate rhizomes were complicated each other. So high density of genet, such interminglement of genet may be important factor for sexual reproductive success in the time of mass flowering. So we move to the last topic, joint evolutionary hypothesis between the rhizome system and the flowering behavior. Bamboos are thought to have evolved from tropic to temperate zones. There is a clear climb of life forms and coronal structure from tropics to temperate. And furthermore, not only morphological features, but also the flowering behavior are quite different between tropic to temperate. Especially temperate bamboos have a quite long flowering cycle over 100 years. To understand the extremely long flowering cycle of temperate bamboos, coronal interminglement of the genet interminglement is a very important factor. For example, in the case of tropical bamboos, too large clone bring about the self-pollination and selfing ratio increased, large increased as genus size increased, like this of the Merocana Baxifera long neck Pacmorph type. On the other hand, temperate bamboos with a complex mixture of genet, selfing ratio did not change regardless of genet size. This indicates that if the flowering cycle or longevity of bamboos elongate longer, high reproductive success may be maintained compared to the clump.
You have 2 minutes left.
Okay, thank you. High reproductive success may be maintained compared to clump-forming tropical bamboos. In other words, genetic intermingled structure in temperate bamboos bring about high sexual reproductive success even after long clonal growth. So it may be an adaptive feature of delayed flowering for temperate bamboos. Such idea is supported by theoretical study as long rhizome drives the evolution of delayed flowering. So we should pay much attention of the relationship between clonal structure and flowering behavior to understand the evolution of bamboo flowering behavior. Okay, the conclusion of today's presentation is rhizome systems and clonal growth patterns are important characteristics to understand the whole bamboo life histories. Okay, I appreciate my many collaborators and Forward Bamboo. Thank you very much for your attention. Sorry for some trouble in my presentation. Thank you.
No problem. Thank you. Thank you, Professor Makita. As we know, bamboo flowering is a misery. So Professor Makita gave us a hypothesis between the relationship of rhizome systems and the flowering behavior. It's a very great presentation. If we have any question, maybe we can make it later on the Q&A section. Okay, thank you very much for your presentation, Mr. Makita. Thank you. Yeah, you can quit your sharing. Okay, thank you. Okay.
Thank you.
Next, I would like to introduce Dr. Sinkong Axiala. Dr. Sinkong Axiala is serving as the Deputy Director of Forestry Research Center, National Agriculture and Forestry Research Institute of Laos.
Yes, this is too convenient. Can you share my video?
Yeah, and today he will sharing with us a topic about the distribution patterns and above-ground net primary productivity on bamboos in northern Laos. Hello, Dr. Sinkong.
Yes. Yeah. Can the host share my video? Too convenient. Can?
Yeah, you can share your PowerPoint now. Yeah. Can you open your video? Turn on your video.
Yes, we can.
Yeah, we cannot see you now. Yeah, great. Now, yeah. Okay, thank you.
So today I would like to present the topic is distribution pattern and above ground net primary productivity of bamboo in northern Laos. So the content is general introduction, objective, material and methods, result, and discuss my research area in northern part. Yes, thank you very much for your introduction. Let's I share the screen. Hello everybody, my name is Mr. Sinkong Axiala from Laos. I work as National Agriculture and Forestry Research Institute. So today I would like to present the topic is distribution pattern and above ground net primary productivity of bamboo in northern Laos. So the in content is general introduction, objective, material and methods, result, and discussion and conclusion. The first slide show that map of Laos. So here in my research area in northern parts of Laos, there are 8 provinces and 47.6% of the land cover area. They located in northern center part of the Indo-Chinese Peninsula, and there are tropical, subtropical, and temperate climates. And so there are maybe diverse bamboo including both of the paleotropical and temperate woody bamboo. And in northern of Laos, bamboo is one of the dominant plants in fallow forest. That is a large quantity of valleys bamboo are likely spreading across. However, even the taxonomic study of bamboo are extremely limited. The species identification of bamboos is still confused, which have hindered future ecology study on bamboo. And this also the bamboo is important for socio-economic and ecological. For the socio-economic, bamboo have been closely associated with local people daily life and used in various ways, such as house construction, traditional bamboo music, and other tools and material. Due to recent development of the engineering technology, the bamboo have been increasingly used in the commercial of products like clay bamboo, lamp, textile, charcoal, and other. And for the ecology important, there's a bamboo have been high potential to sequester carbon. Rehabilitation of the degraded land, soil erosion control, and great conservation for significance. Above ground biomass and net primary productivity. The global estimate for above ground biomass of bamboo total 32 to 256 mg per hectare, and above ground bamboo net primary productivity 7 to 48 mg per hectare per year. In northern Laos, there are 2 previous studies on above ground biomass, but no report on above ground net primary productivity of bamboo. Reliable allometric equations are needed to accurately estimate biomass and net primary productivity. And so my objective like the specimen edit and how to identify by Dr. Sarabut Sankel from Kasitsat University, Thailand, Professor Sia and from South China Botanical Garden from China, and Dr. Jakapong Rattan from Biodiversity, Thailand. For the other aromatic question, I collect that is 11 species of bamboo, and also analysis by between the component and interviewed by Dr. Niruta. The standard of aromatic equation. April 2017 to March 2018 in Rattan province, and also annual or monthly above ground metal productivity also calculation. The result and discussion. I found that the total of the bamboo species up to now is 63 bamboo species, including 17 genera, and 13 species were unidentified.
You have 2 more minutes.
And there are 8 species were new records now, and some of 4 species.
Maybe you can skip some slides. Yeah, thank you.
Bamboo species identification. For wide bamboo, there are wide distribution among species. Yeah, so that is S1.4, Thailand and Vietnam. Environmental factor associated with wide bamboo. Some species near is NNDS conculated the bamboo lining to skip to the evaluation and connect. That's bamboo common evaluation. So the specific piece, the subspecies specific tolerant to low temperatures may be underlay the transition of observed. In my study, the composition multi-species and so the aromatic equation by DBS and DBS and I. A little bit DBS and I is higher than DBS, but I recommend that aromatic equation in DBS is better, because if we need this one, we take time to collect evaluation biomet, even in study. So, Sympodia bamboo, Sympodia bamboo is rainy season and dry season of the primary productivity. That means as new calm requirements in the response change in precipitation or air temperature. The future climate change may impact the carbon sequestration rate in bamboo. In conclusion, truth bamboo inventory in national taxonomy information was qualified. So, yeah, bamboo species in northern Laos, and to compare between the Asia Pacific region, total number of bamboo species now 121 species. That means it's a high carbon sequestration potential of bamboo was indicated. And here so is the above ground carbon stock and in northern of Laos, like this, and also above ground carbon fission in follow of northern of Laos.
Okay.
Perspective from the future study. The future
Thank you.
inventory is needed to produce the complete species list of bamboo and clarify bamboo distribution pattern in now. And
Thank you. Thank you, Dr. Axelaya. Yeah, thank you very much for your sharing. And yeah, you give us the introduction about the bamboo productivity in Laos. It's very important basic data for calculating the carbon sequestration capacity of bamboo forests. Thank you very much. Okay, due to the time limitation, we will move to the next speaker. Associated Professor Ge Xiaogai from the Research Institute of Subtropical Forestry, Chinese Academy of Forestry. She will share with us physiological integration of carbon and its dynamic response to drought stress on bamboo, Mosul bamboo plantation. Dr. Ge, you have 12 minutes. Yeah, you can share your PowerPoint now. Yeah, go ahead.
Good morning, ladies and gentlemen. Could you hear me?
Yes, very clear. Yes, very clear.
Okay,
yeah, go ahead. Yeah, thank you.
Okay, thank you. My name is Ge Xiaogai. I come from Chinese Academy of Forestry. My topic is the physiological integration of carbon and its dynamic response to drought stress of most bamboo plantation. My topic includes 4 contents. First one, I'd like to introduce my research background. Everyone knows that climate is global issues for the international community at present, especially climate warming and precipitation patterns are 2 important features of climate change. And the global precipitation will show a trend of the drier the dry and the wetter the wet. From these figures, I'd like to introduce from the photosynthesis and to soil. It is ensures our path and especially insect fair and withdraw. When each drought, maybe it can shoot hydraulic failure and carbon starvation until the tree is died. So the drought has a significant impact on the whole cycling process and the carbon sequestration potential of a terrestrial ecosystem. This picture also studies the carbon allocation and the drought. Drought affects the amount of new carbohydrate, the distribution, uptake, and utilization between different tissues and organs. Influence factor of different tissues and organs in urban and battleground are different. This is also new carbon allocation in newly produced carbon and non-structured carbon in different functions. In drought, some carbohydrates are used as a chemical defense to protect the resources that has been acquired. The plant also exports pet carbon to the soil in the form of root elucidation to increase the nutrient uptake. This figures shows and drought condition. Maybe the trees or plants followed negative carbon neutrality. When this is under condition, photosynthesis and growth are down-regulated, while sucrose and starch, both synthesis pathways are upregulated. Trace phases serve photosynthetic limitations, strategically regulate storage and consumption at the apex of growth. This figures show that and drought stress, the new carbons will have a great change in quality carbon uptake, carbon substrate, and substrate allocation, zinc activity, especially the new coverage allocate respiration and growth. The portion has great change. I'd like to introduce different species have different response. This is after labeling 8 days. The new carbon of the coniferous tree species is distributed 30% to the ground, while the broad-leaved trees species below 10%. This is also see different species, bare mass change leaf and respiration and so on. So the 13 carbon was remained 4 days in leaf of carnivorous, but just only 1 day in brown leaf trees. Different species could optimize the portion of new color calibration and ground and below ground. This picture different the effect of drought on change of NFC in different against. Drought treatment increase the alum water potential and phloem solution concentration. And normal condition, the phloem promotes the transport of water and carbohydrates in the alum. That in and drought condition increase the solution concentration of the phloem, prevents excess water loss in the alum with a positive effect. This picture shows the different tissues and organs have different response. Include primary metabolism and secondary metabolism. The study showed that drought stress forces starch into soluble sugars, and a large amount of accumulate soluble sugars affect photosynthesis. So C accumulation and component change affect plant growth. Studying C accumulation and distribution patterns in different plant organs contributes to a more comprehensive understanding plant drought adaptation strategies. This is the second part. I'd like to introduce my scientific questions. In my experiment, we select a 1-year-old mossel bamboo and labeling it. Then we want to investigate the new carbon carbohydrate transfer from the mother bamboo and grandmother bamboo. The question is two. First one, does new carbon allocation follow a distinct priority or demand priority and drought treatment? What are the distribution characteristics of photosynthesis products and the sharing mechanism between plants and drought treatments, and its key influence? In the moss bamboo growth process include off-year and on-year. The study would elucidate the mechanism of drought stress on the physiological integration of carbon on moss bamboo and reveal the mechanism of change relationship induced by environment stress. This is our study site. located at Qianjiangyuan Forest Ecological Station, Hangzhou Province. And the experiment year of most bamboo plantation is about 30, and the density standing bamboo is to 2,000 to 2,005 plants per. This is our design finger. The simulated drought was carried out in situ by the seeding method to study.
Maybe you can more quickly. You have 4 minutes left.
Okay, the new carbon allocation and resource share mechanism on Mosul forest. This was 6-year-old Mosul bamboo, and then sampling were several days after labeling. This is our results. Our results showed drought decreased the 13 carbon peak value of leaf, branch, and root greatly. Drought reduced new carbon uptake in Mosul bamboo without affecting the rate of new carbon transport. Drought increase also affect soil atomic press 13 carbon. There is no 13 carbon single in the soil after labeling 90. There was no difference between drought and control after 30 days of labeling. We also found the new carbohydrates in mother bamboo and grandmother bamboo. So our results confirm that the new carbohydrates enter the mother and grandmother bamboo through the wave root system. The new carbon allocation of Mosul bamboo follows a priority of demand rather than a priority descent.
You have 2 minutes left.
Okay, this is the conclusion and the discussion. The proportion new carbon in root system was end stage non-destructive sampling. This is other results from the NSC. Other results support verified in our study. There was no significant difference of soil and drought in study, so the new carbon transfer rate was not an effect. Drought treatment increased below ground carbon allocation. We also showed other research about drought. For example, the effect of drought affect late fall and later decomposition, later degradation decreased greatly, and drought decreased the contribution of later quantity to soil nutrients that increase the later quality and the soil macro community to soil nutrients. We also researched the effect of drought on the soil respiration, especially later resource contributions. Later removed decreased the soil respiration, and later doubled increase the cumulator CO2 emission, and later removed decrease greatly. That's all. Thank you.
Thank you, Dr. Ge, for your sharing. It's a great research. Use the carbon-13 to trace the carbon allocation to these colonial plants, that is bamboo. I think it will give a great and a very important implication for the further understanding of on the how bamboo respond to the drought stress. Thank you very much. Okay.
Thank you.
Due to the time limitation, I think we only have 1 or 2 small questions. Is there anybody have any question to the speakers? if there is no question, I think I would like to thank you very much for the guest speakers for all your great presentations. Now I would like to invite Professor Wang Guangyu from University of British Columbia. He's serving as the Associate Dean of Faculty of Forestry. He will give us a very quickly concluding remarks. Professor Wang, go ahead.
Yeah, thank you so much. Thank you so much, Jinfei. Good morning, chairs and excellent speaker and participant. This is my great honor to deliver this closing remark. First of all, I would like to thank all the organizers, particularly for ICBR, for organizing this kind of wonderful events. And secondly, I would like to thank all the speakers, it's excellent speaker, for the talk today, which inspired me a lot, not only for diverse topic we have around the carbon and the carbon forest, but also it's the depth of the research. So bamboo is widely distributed globally, and its unique biological feature enable bamboo contribute more on the other species on environmental and local community economic developments. So bamboo has a higher carbon benefit, such as carbon sequestration. Bamboo forests have a short rotation age, and they have a high ecological and a social benefit. So today we have that 6 speakers. They are talking about bamboo from different aspects, from urban setting, from the rural community, from bamboo scientific like a flowering mechanism, and also the response of the bamboo to the climate change. So it's very excellent talk, and I hope this conversation can continue on in the future. Particularly right now, we are facing a lot of challenges, particularly like a bamboo carbon market, bamboo labor shortage, and also the climate change is increasing the challenge for the bamboo and the bamboo development. So I hope we can continue this kind of discussion to lay out our best practice for bamboo development, bamboo forest development in the future. Thank you so much. Thank you, Jinfei.
Thank you very much, Professor Wang. Yeah, I think due to the time limitation, I think our session is now come to close. I would like to thank you for all your participation, for all the guest speakers, for your great sharing on your research. I would also like to thank Dr. Peng for her moderating. Thank for Professor Wang for his great concluding remarks. Thank you all for your participation. If you have any questions, you may by typing on the dialogue through the Zoom. Okay, thank you very much. We have to close our session now because we have already run out of time. Thank you very much. Thank you.
Thank you all. Thank you.
Thank you.